Good morning students, welcome to the biology class. Today we are going to discuss about the Cilia and Flagella. Cilia and Flagella are hair-like movable projections outgrowth of cell membrane. Cilia are much smaller but more in number, while flagella are larger than cilia and few in number per cell. Cilia may help in the movement of the cells bearing them or the movement of the fluids and substances inside the organ, while flagella help in the movement of the cell bearing them. In this image, you can see the small hair-like structures, that is the outgrowth present on the cell membrane of the paramecium, which are small in structure but large in number. And in euglena, you can say that is few, that is one only, that is a long structure that is known as your flagella. So they helps in movement as well as that helps in moving the substances if they are present in some organ, and this helps only in movement of the substances. So in this way you can say that flagella or the cilia is being divided into the four parts, that is your basal granule, then you are having a basal body, then you are having the shaft or axonemia, and when we cut the cross section of these you will get the TS of the flagella and cilium, which will be explained in our further slides. So the ultrastructure means seen under the electron microscope shows that cilia and flagella are membrane-bound cylindrical structures having four parts: rootlet, basal body, basal plate, shaft or axonema. Cilia and flagella are attached with the help of a rootlets, and when we see the transverse section of the axonema or the shaft, it is like that. A basal body lies at the base of each cilium and flagellum in the cytoplasm. It shows the 9 microtubule triplets along the periphery, that is 9 plus 0 organization. The basal plate separates the axoneme from the basal body. Axoneme possesses 9 microtubule doublets along the periphery and a pair of centrally located microtubule, that is 9 plus 2 organization of microtubules. The central tubules are connected by bridges, and they are also enclosed by the central sheath, which is connected to one of the tubules of each peripheral doublet by the radial spoke. The peripheral doublets are also interconnected by the linker. Each doublet also shows a pair of arms made up of proteins, that is a dynein. Cilia and flagella, they are able to bend because the adjacent microtubule doublets slide over one another while maintaining their individual length. So in this image you can see this is the transverse section of your cilia, that is about your basal body. So you can have 1, 2, 3, 4, 5, 6, 7, 8 and 9 peripheral microtubule, that is doublet. In the center you are having 1 and 2, that is a central microtubules. So that is 9 plus 2. This is 9 plus 2 organization. So central one, they are being surrounded by the central sheath and linked by the bridge. Each peripheral microtubule is being attached with the center with the help of, that is the radial spoke, and they are having their own linkers, which is made up of a protein, that is the dynein. So this is your 9+0 organization, cilia and flagella, made up of a peripheral microtubules as well as the central microtubules, and this is being surrounded by the membrane, that is your plasma membrane. So this is very important that you have to explain the ultrastructure of cilia or flagella, that is 9+2 organization. So the next organelle, that is your centrosomes and centriole. So centrioles are a pair of darkly stained granules found in all animal cells except mature mammalian RBCs, that is should be in the capital, prokaryotes and higher plants, but they are absent in prokaryotes and higher plants. Please make the correction over there. They are found in all animals except mature mammalian RBCs, prokaryotes and higher animal. Means they are absent in RBCs, they are absent in prokaryotes as well as in the plants. They always occur in pairs perpendicular to each other, and these are also called, that is diplosomes. So that is another name of your centrioles. They are inside the specialized cytoplasm called as the centrosphere or kinoplasm. So the complex formed of centriole and centrosphere is being termed as centrosome. Structure. Each centriole has a cart wheel-like organization with 9 evenly spaced microtubule triplets along the periphery connected to the central proteinaceous hub by the radial spokes, also made up of proteins. The adjacent microtubule triplets are connected by Ca linkers. The triplets are being named as A, B, C from inner to outer. All the microtubules in centrioles are formed of protein tubulin. There is no membrane boundary for the centriole, but it is surrounded by pericentrioidal satellites, which are dense amorphous protoplasmic sphere in one or more series. In this image you can see this is your animal cell, because centrioles or centrosomes are present only in the animal cells. They are absent in the plant cells. So the location of the centriole is just above the nucleus. So these are two granule-like structure which are being known as a centriole, surrounded by the cytoplasm, that is your centrosphere. So centrosphere plus centriole, that is called as your centrosome. And if we see the crossverse section or the transverse section of centriole, then the picture come like this way. So this is your pericentriole satellite, because this organelle is without the membrane but surrounded by the pericentrioidal, that is a dense amorphous structure, and they are having a 9 triplet microtubule, that is your 1, 2 and 3, 1, 2, 3, 4, 5, 6, 7, 8 and 9, and they are being connected to the center, that is your hub. It is again made up of protein, and that is having A, B and C, and they are linked by AC linker. So this is A, so this must be the C, that is A, B and C, and that is called as an AC linker. These are the triplet fibers, and these are the 9 radial spokes. So they are being attached, and along with them they are having the XY linker. So that gives a, you can say, the appearance like the cart wheel, like this way they are being linked. So that gives the appearance like a cart wheel. So that is the structure of a centriole. Again, it is very important. The function is centrioles form the basal body of cilia and flagella, and they also give rise to the spindle fibers during the cell division. So that is all about your eukaryotic cell. About the organelles, we have divided the eukaryotic cell into three compartments, that is the outer membrane. Along with that, we discuss the plasma membrane, cell wall, then the inner one, the nucleus, and then cytoplasm and in organelles. In the cytoplasm and organelles, we discuss about the endomembrane system, mitochondria, plastids, cytoskeleton, ribosomes, various microbodies like spherosome, peroxisomes, and very important, the outgrowths of your cell membrane, that is your cilia, flagella, and non-membrane organelle present only in the animal cell, that is your centriole or centrosome. So by this we finish this chapter, that is a cell, a unit of life. So please go through the NCERT text questions, revise your chapters, and we will have a queries in our next session. Till then, stay home, stay safe, stay healthy. Goodbye. Thank you.